Start recording. Great, we're recording. So hi everyone, and welcome to our second installment of the Spring into Science Queer Representation and Research Seminar Series. Today we would like to welcome Jouvay Pantophlet, who is a self-proclaimed professional pooper scooper. They've been lucky enough to be a researcher of freshwater turtles, sea turtles, birds, and mice, as well as a zookeeper and a certified dog trainer. Currently, they are a laboratory animal technician at the College of Staten Island in New York City, and their master's research focuses on improving the biological relevancy of the facility's 7+ naked Molerat colonies. Just a reminder, if everyone could please mute themselves during the duration of the talk, that would be great. And if you have any questions for Jouvay, we'll have a short Q&A session afterwards. So without further ado, I'd like to pass it along to Jouvay. Thank you so much for joining us today.
Thank you everyone for having me. Let me just start sharing my slides. Please forgive me, I am not a Zoom person. Okay, share this. Okay, here we go. So this is my master's project, and it's also a personal project because this is dealing with the animals that I work with on a daily basis. So the working title is Increasing the Biological Relevancy of Naked Molerat Enclosure. So it is by me, Jouvay Pantophlet, and my advisor, Dr. Dan McCluskey, who is also my coworker. So you'll see me talk about him as without the honorific and just say Dan, because we have that relationship. So some lore. So Dr. Dan McCluskey is an associate professor of psychology at the College of Staten Island. He is a recipient of the Presidential Early Career Award, and he was awarded it from President Barack Obama. I think back in, I want to say 2015. I'm not sure on that, but I can check on that. And he really likes naked Molerats. Like, the dude really loves naked Molerats. And I, Jouvay, am a professional pooper scooper. Previously a zookeeper for about 4 years at the Staten Island Zoo here in New York City, a dog trainer. I'm currently in my master's at Hunter College, which is also a part of the City University of New York conglomerate, for animal behavior and conservation. So kind of melding all of the things that I've done as a zookeeper, all of the things that I've done with conservation together in one degree. And I also really love naked Molerats. So what in the world is a naked Molerat? Because whenever I say I work with them or I'm doing research on them, people are like, okay, but what is that? So the naked Molerat is a wonderful little being. It is the longest lived rodent in the world. It has about a maximum lifespan of about 30 years. In the literature, they're called saber-toothed sausages, which I adore. They are known, they make their steak for a couple of reasons, but in the biomedical world and the biological world, they are famous for being one of 2 eusocial mammals. So like your, ant colonies or like your beehives, they have one queen. Unlike a beehive, that queen is chosen not through birthright, but through sheer force and conquering. And they have the same sort of caste systems. They work in that manner, and it's phenomenal. Their closest cousin, the Damaraland Molerats, which are basically these guys but with fur on them, are also eusocial. So they are very, very distinct class, and they're neither moles nor rats. They're their own little, corner of their taxa. Their senses are absolutely amazing. So they are naked, but as you can see probably in this picture over here, they have these little whiskers, and those whiskers run along their body in rows. And kind of like a Venus flytrap, if you touch one of those hairs, they will orient to that place. So they have this really amazing somatosensory system going on. Most of their brain innervates in their teeth. So current literature thinks that they have this whole sensory world through the feeling of their teeth, which is amazing. They are not really great with the eyesight. As you can see, they're kind of these little beady dots. So they can sense light. Doesn't seem to be all that great. Aside from that, we don't really know much about their vision. they're also unique because they are, I'm going to say this wrong, but I'm going to try, poikilothermic. So they are mammals, which makes them, endothermic and warm-blooded, but they are subterranean animals, and they're hairless, and they also have a unique social structure. So they kind of are dependent on their environment for their range of temperatures and what temperature their body is. So they'll thermoregulate more so through their behavior of like doing these like cuddle puddles. And you can see here, it's just a mass of them in a little puddle. Or choosing vertical space in their colony as to, how much heat they want. So closer to, above ground, obviously the warmer it gets, and the lower down, the cooler it gets. So that's how they thermoregulate. They are herbivores. They eat geophytes. So geophytes are basically like your tubers, succulents, anything with the, big taper roots or things that store a lot of nutrients and a lot of water, because they don't drink water. Like you'll never find them like near a puddle. So they get all of their water and all of their food at the same time from their food source. It's believed currently that their eusociality evolved because of the sparsity of their food sources. Since they live in subterranean areas in dry grasslands from like southern Kenya down to South Africa, their food sources are very sparse. It's kind of scrublandy. It can be, super dry to even desert, and very sandy. So those plants aren't growing, very close together or numerously. And so it makes more sense then in that sort of context to have a bunch of animals that delegate their time and their energy efficiently, instead of everyone trying to find the food sources for themselves. Delegate it to a few scouts and a few workers that if they find a good geophyte source, they can go then and focus on that. It also seems like they farm their food, which is really cool. So they eat just the center of it, and then leave the outer ring so it kind of regrows back and they can go back to that food source. So sustainable farming, yay. So these guys are even more cool in where they live because of their adaptations to those things. So they can live in loam soil, which is kind of a light, mix that's porous. It's got room for water. It can be sandy. They can live in laterite, which is a very red, rich, sort of almost clay-like. It can be light and airy, or it can be basically compressed into bricks, or they can live in pure gypsum, which is kind of like saying they can live in like cinder block. And they can do this because of those giant teeth. If you're not familiar with rodents in general, rodents have ever-growing teeth, so they will grow throughout their lives. And I think for these guys, it's a wild growth rate that they have. And if you can see over here, their teeth erupt before their lips. So when they're digging, they're not swallowing and ingesting things that they are digging. They're just eating through and then spitting it out behind them. And, you'll see in some videos, they kind of like paddle all of the material behind them. So it's super cool. They're studied, they've been in the biomedical laboratory field for about the past 50 years. And they're studied mainly because they are really weird in how they survive. They are basically cancer resistant. They can get cancer. It's not like it's, unheard of, but they are super cancer resistant. They can survive in 0% oxygen for over 30 minutes. They kind of just pass out a little and then get up and they're fine. And it seems like whatever's going on with them prevents them from having brain damage, which is why they're studied for things like epilepsy, even like Alzheimer's or any neurodegenerative diseases. They have negligible senescence. So basically, you kind of don't see them. I mean, they already kind of look like little old sausages, but phenotypically, you don't really see anything happening. And if I'm remembering correctly, on a cellular level, it's also very hard to tell how old the animal is. And again, they can live for 30 years. So you just basically find them passed on, and there's kind of no warning to that, unless there's a fight in the colony or something. So that's basically why they're mostly studied in a biomedical sense. So we study them mostly, Dan studies them mostly for behavior. So we have a setup that kind of just, lets them be. So our current setup, we have about 7+ colonies. And I say plus because we have some breeders that are currently founding a colony, and it takes a really long time for them to do that. So we have like 6 to 7 full-fledged colonies and a couple more that are just starting up. So I'll play a video for you so you can see as I explain this. You'll actually see a queen rush in from the side, and she's bulbous and huge and very pregnant. There she is. So what we have is we have these sort of chef boiler pans, these very heavy PVC pans that you can see are laid out sequentially adjacent to each other, basically across a whole room. So each level is one specific colony. And each chamber, we call them chambers, is connected by a bunch of PVC piping. And then what we do is we put corn cob, it's shredded corn cob, which I've been working at my current facility for about 5 years. And they would always say, oh, it's cob, it's cob, it's cob. And I never put it together that it was actually corn. It's actually corn. They take the corn cob after they've taken all of the edible parts and they put it in a shredder. And then this is what you get after it, a really great substrate, usually for mice and rats in a laboratory setting. And we put that in, and basically they get to do whatever they want with it. So as you can see, they have a food chamber. So over, you could see the end chamber here. So the chamber right over here, we make sure to feed them in the same exact place. And I'm saying this now because you'll see eventually why that's important. So we will feed them basically ad lib. We go in and refresh them about 3 days a week and make sure that they have all of their food. And as you can see, we usually cut it up. Sometimes we'll leave it whole for them to chew through. And then we just put it in there. And if it gets moldy or anything, because their microclimate is very humid, we just take it out and replace it. About every 2 weeks, they get changed over, which means that we take all of the Molerats out, we disassemble all of that, and then put together new pieces that are cleaned, and put the other stuff through the cage washer. We have a giant cage washer, and if you've never experienced a laboratory facility cage washer, we have a walkthrough one. So it's basically these 2 giant doors. You close one side, you load the stuff in, and you close the door behind it, and it goes up to about 185 degrees Fahrenheit to basically heat and kill everything. And we use heavy detergents to make sure that everything gets washed away. So this is our current setup. And as you can see, we use rocks on the lids because naked mole rats are really great at trying to escape. And we've had a few escape events, which I can talk about in the Q&A after. Okay, so some of the issues with this is the enclosure is designed more for convenience. And I should point out that from our knowledge and what we know about how other facilities, aside from mainly zoos, but how other laboratory facilities care for their mole rats, we have the most unique setup and the one that is so far closest to their most natural environment. So, but saying that, their enclosure is still designed for convenience, more convenient than what it would be. But it is arduous to service it. It takes a full 2 days to do so. So you take all the mole rats out, you take all of the caging apart, and you have to twist it, which leads to carpal tunnel. And we've had 2 techs already have to have surgery for this issue. You have to undo these very tightly screwed PVC, hand-threaded PVC parts, put them in bleach to soak for 24 hours, scrub them out, then throw them in the cage washer to get the bleach removed, then also put together the colony and then introduce the mole rats to that colony. So it takes about a 2-day period to do all of this from start to finish. And it takes the techs who are servicing them, it takes them a full day of working on the mole rats and a full day because we also have it at a very high temperature. So you have to take a lot of breaks. So they can't just like go in, bang it out in like 3 hours and then be done. So it's a lot. It's a lot. So on the tech side, that's the issues that we have with it. On the research side, Dan is studying them for their natural behavior. And we know that they are great architects of their environment. I mean, their teeth are absolutely set up for it. They're adapted to dig to create their own environment. They lack that choice and control in what we have for them currently. We already have these pre-established chamber sizes. They have a pre-established path. They also can't do any of those thermoregulatory, changes in depth. So they have no vertical space to really do anything with. And they also don't have any of the substrate that they would normally find in their environment. So they're not really digging through anything. They're not using those teeth, which on, a natural research, natural behavior level is not the greatest, but also on a husbandry level. Remember how I said that those animals, rodents in particular, their teeth are ever growing. If they don't use them, what happens is they get all gnarly and corkscrewed and can even, erupt into their skull or through their skin, and it's not really pretty. And so then you have about 70 plus animals per colony that have these gnarly, snaggly teeth that you have to go in with basically toenail clippers and clip off their teeth. And that adds to their husbandry. I've had to do it for a couple of animals, and it takes a couple of hours because you want to be, you don't want to stress them out, you don't want to accidentally injure them, and it's a very sensitive place that you're trying to cut. And as I said, their whole world is through their teeth, so you have to be sure that you're not, going near any live tissue, and you're just making sure that it's the parts of the teeth that are ready to be shed. So it's a delicate thing to be able to do. And then, as I was saying before, their eusociality evolved most likely because of their sparsity of food availability. So if we're giving them food, as I said before, all in the same chamber, and we're giving it to them ad lib, and they're not going out and looking for it, what are we doing to their selective pressures? Are we compromising their adaptations and therefore their evolution, and therefore, are we going to be able to see their natural behavior? So these are a couple of the issues. So it got me thinking into, how could we fix this? What could we do? What could we do to increase that biological relevancy? So when I say biological relevancy, I mean a situation that they're in that is more relevant to their adaptations, that is more relevant and represents and reflects those selective pressures that brought them to be where they are now. And granted, you can't fight all of them, because not only do we not, know what led Molerats to evolve to be Molerats in full, but we also don't know how other issues are going to selectively affect them in the future. But there's no reason to just be like, put our hands up and say, all right, well, they're just gonna go on a path and we can't see it, and I guess we'll deal with it when we get there. We should try and be proactive about it, especially because these animals are so new to the laboratory field. Again, they've only been in laboratory facilities for about the past 50 years, possibly less. We should be proactive. We should make sure that what we're doing is the best that we can possibly do. In animal care, in the husbandry world, we have a saying that there is no such thing as tradition in animal care, because you should always be up to date on your methods, and you should always be trying to improve, because your quality of work is their quality of life. So with that, I started doing a literature review on previous works into Molerat enclosures, because if you're from the zoo world, or if you have experienced researching any other animals, like pick a chimp, any apes, any monkeys, big cats especially, you can find mounds of literature about what types of enclosures work for them. This is the complete list of 2 years worth of me trying to find husbandry related literature. It's 5 articles. Not great, right? So we're studying them. If you just type in naked Molerats on Google Scholar, just a cursory glance, you will find mounds of literature on their ability to survive in 0% oxygen, their ability to not get cancer, all of this biomedical research, but no one is just looking at who they are and what they do in their natural setting for what they are worth. So that was slightly depressing and slightly alarming, because now we're studying these things and we don't even know base one of them, right? Okay, so I looked up all of this, I got very depressed, and I thought, all right, what can we do? So and given all husbandry challenges, given the natural history, is there a better way? What in the world could it be? And I have been someone in my own personal life, I have wanted to downsize and live in a tiny house, and so, or in a sustainable sort of way of living. And so I was someone who was always researching, what are sustainability practices, permaculture, all of that stuff. And in those circles of green living and tiny house living, there is something called Papercrete. And Papercrete is basically exactly what it sounds like. It is paper concrete. So it is any sort of material, construction material that's any sort of pulped paper mix mixed with cement. So I'm going to make a distinction here because I had to learn this too. So cement and concrete are technically two different things. So cement is what makes up concrete. Concrete is just this heterogeneous mixture of things that are bound together by cement. So Papercrete is a concrete of paper and Portland cement. And it is so much less dense than traditional concrete by, and you'll see in my own research, by a ridiculous amount. We got it up to about 60% less dense, but still has the same amount of strength and integrity as a similarly sized piece of traditional concrete, to the point that if you took one of these blocks that I've made right here, it can hold up a car just as well as, you know, the traditional similarly sized block of traditional concrete. Amazing. And it's super easy to make because, again, you know, Portland cement is readily available, at least here in New York City. Any Home Depot, any you know, home improvement store has at least a 90-pound bag of cement just laying around. And paper, well, that's a great thing we can easily source. So we had to look into a couple of different ways and a couple of different materials. But what we found, and this is something that I neglected to put in the PowerPoint, I apologize, was that newspaper was going to be the best for this because most of the newspaper in the United States, the ink is soy-based. And there's studies where they've just pelleted up newspaper, fed it to cattle, and it passes right through them. And they've done toxicological studies, and it really doesn't have much effect. So we figured that newspaper would be the best, as opposed to, you know, just taking any random sources of, you know, paper that was from envelopes or documents, printer paper. We didn't know what was in them, what was used, but we know that newspaper is generally a safe material to use as not only a bedding, but apparently also as feed. So my methods overview: figure out how to make the stuff. How do you make the stuff? It's basically a bunch of ragtag, like, you know, tiny house builders and green builders just kind of being like, oh, you take a bag of this and some of that, and you do some of this, and you mix it together, and then you get Papercrete, which is okay, great for, you know, if you're, you know, messing around and building your own house. But in a scientific setting, I need a formula, so I need to figure out how to make the stuff. And then we wanted to track how much weight will it lose from pouring it and casting it to when it's fully cured. So cement needs at least 28 days to cure. And that is, curing means that it goes through the full chemical process of hardening from when it was mixed with water. So it takes about 28 days, and then you can do stuff with it. So how much weight will it actually lose? And that'll be important, and I'll explain further down the line of this presentation. And then we wanted to look at, okay, so if, you know, we have these blocks, let's say it all works out, it's all great, we cast them, they're pretty light that we can handle them. If we stick them with the Molerats, are they going to be able to survive this microclimate that they create? And the Molerats create a very humid, very hot, and they live in a very hot room, and they make it even hotter by cuddling together. You know, is this thing going to grow mold? It's part organic material. Is it, you know, going to be something that just degrades and melts? Like, we had no idea what was going to occur when it was with the Molerats. On top of that, you know, is it going to last? So we do a changeover, which means we take apart and do that whole servicing. We do that every 14 days. So is this going to last to at least the same amount of time? Because it wouldn't make sense to put in something new that takes more husbandry opportunities, right? We don't want to increase the amount of time that we're doing things, especially because we want to leave them alone to do their natural behaviors. If we're always disturbing them, that defeats the purpose. So will it last 14 days? Will it not? Who knows? And then what do the Molerats think of this stuff? You know, kind of like a focus group. What are they going to do? What do they think about it? Are they going to eat it? Do they think it's going to be food? can we confirm that they're not going to eat it? You know, we know evolutionarily they have an adaptation to make sure that they're not going to eat the stuff that they're digging through. But does this look like food? Will they try it? Are they, you know, neophobic, that they don't like any new things? What's going to happen? So this is the process. And I am not going to play it with sound because it's got a bunch of drilling. So you take a bunch of paper, you put some water in it, you let it soak for a while. Then, and this is my housemate and best friend, Rachel, you take some, let me go back to that actually. You take some of it, and what you do is you take a drill with a paddle on it and you drill the crap out of it until it's basically a pulp. We found then that afterwards you needed to take out a lot of the excess water. So I basically put in a cheesecloth, kind of like in the way that you would make mozzarella cheese. Put it in a cheesecloth, and as you can see, it's very soupy. And then I, you know, squeezed it out as much as possible. And then what we did is we mixed it with the cement. We put it into these silicone ice cube trays. And this was just a test, like, what are they going to do? What's going to happen? And then about 24 hours later, it was set enough that I could pop it out. And this was the first unveiling of them. I was very excited. And it worked. It was there. And then we set them to dry, and I made sure to have little labels for the recipe. And then what we did is we casted a giant chamber that they're already in. We casted that giant chamber and let it, you know, obviously you could see all the water that it's already losing, and let it cure for 28 days. The reason that we did this is because this is Anna. This is my coworker, Anna, and she is one of the primary Molerat keepers. She doesn't lift. The most that she can lift is a grain bag, as you see right there, and the grain bags weigh 50 pounds. We wanted to make sure that if that's her maximum, anything that we're building is going to be that or less. And knowing that this material is supposed to be way less dense for its size than traditional concrete, we're like, okay, let's test it as big as it can go. So what we did is we tracked the weights. We started with, we call it a small chamber. So we have these little chambers too for the Molerats. So we did a small chamber, a large chamber, and then those 2-inch by 2-inch blocks. And we tracked their weight and how much weight they lost each day. So you'll see the first two are in pounds, and I apologize. And then the last column, the 2-inch blocks, are in grams. So we started at 47 pounds for that large block over there. And we got down to, after about 30 days, 17 pounds, 17 and a half pounds. So it went from being something that you could really barely lift well, also because it's clunky, to something that Anna would take and basically hold above her head and run around with. So cool. First experiment done. We found out how to make the stuff, and we tracked its weight, and so far so good, right? This seems to be more of a viable material. Okay, but now we have to put it in and see what it does in the microclimate. So we did a microclimate test. So what I did was we put it in, and you know, the microclimate test and the Molerat focus group test basically occurred at the same time, because we just plopped it in with the Molerats and kind of recorded to see what would happen. So we put these blocks in, and as you can see the day 1 versus the day 14 and or more in each of these pictures. So you'll see Linus, which is one of the names of the colonies, the test colonies. So Linus colonies block, and you'll see Terrific colonies block, day 1 versus like day 14. And you'll see that, first of all, they had differential interest in it. And the weights differed too from, you know, when we started to when we ended. So it went from, for Linus, 86 to 63 grams and 88 to 84 grams for Terrific. And it lasted more than 14 days. This is just the first 14 days of results, but it lasted more than 14 days without getting moldy, without degrading. The only reason that it degraded was because the Molerats were actually gnawing on it. And so once we realized that they were, you know, actually interacting with it, we started recording them, mostly to just test the equipment. And it was supposed to be really informal. Just, you know, just let's see what happens. It wound up turning out to be the full extent of my research as of right now, because of the pandemic. So this was maybe December into January of 2020 that we did this experiment and recorded them. And we recorded them both with the testing, the blocks in the testing chamber, and then we would record them for a weekend without, just to, you know, have something to look at, like, do they do anything? It was supposed to be informal. I'm just going to put that out there. Um, so once the pandemic hit, uh, we went into lockdown, as everyone in the world did. And what happened was we put all of the animals within our laboratory facility were put on a holding protocol, which means that no research could be done on them. And we didn't know how long this would last, as everyone was kind of wondering how long is this going to be for. On top of that, we didn't know if the Molerats could get COVID. So we definitely were not going anywhere near them to do any sort of research. And if we happened to even just record them and use that for any sort of scientific endeavor, we could have been shut down as a facility. And plus, we were keeping everyone safe, and having to, do a lot of laboratory facilities, unfortunately, to say downsized. And by downsized, I mean culled a lot of their animal population. We had to make the choice to either continue through this very scary situation, or to cull our populations so that way we could remain more safe. We could not bear to do that to our animals. And so we chose to just put them on a holding protocol and just focus on taking care of them every day. So we haven't been able to do research up until about maybe last October. They were on a holding protocol. So most of my research is just these informal videos that we had that I didn't expect were going to be anything. And then, the world had different plans. So we took a bunch of videos of them, and this is sped up. So obviously you could see that they're interacting with them. So what I started to do was I coded the videos using a program called Loopy, which is a highly recommended program. It's a paid service, but it's really intuitive and really user-friendly. And for me, as someone who is dyslexic and has a lot of issues learning disability wise, it made sense for me to use something that was intuitive instead of something that, a lot of academic programs are not user-friendly and they take a lot more to learn. And I wanted something that was easier, because the main purpose was, even though this was for my master's, this was also to make sure that the animals were living their best life. So I wanted to do this as quickly as possible. So we ended up not being able to really see a lot into the chamber. Obviously, we could see that they're interacting with it, but I can't tell, are they using their paws more? Are they biting it? How many mole rats are doing what at what time? And we don't have any physical identifiers. They all kind of look the same aside from their size. So it would be really hard to do focal sampling or scan sampling with them for their behavior specifically. So what I ended up doing was looking at the duration of the animals in the testing chamber versus depending on the treatment. So if it had the block in it or if it didn't have the block in it. And at the same time looking at, well, if they're in the chamber, how many mole rats at one time are in the chamber? And this is important because in nature, they will send out a scout that looks for, just like bees, that looks for a source of food or something interesting, comes back to the colony and recruits. So I wanted to see, do they recruit? So what we found was basically so far, there's really no difference between the treatments in terms of how long do they spend in the chamber. As you can see, it's a pretty even split that they spend in the chamber, out the chamber, depending on the treatment. It really doesn't matter. I haven't really done much statistics on this, but so far, no statistical significance in any of looking at the occupants, looking at, how much time they spend. But what we do know is that they don't ingest it, because we caught it on video of them biting into it and then taking their paws and wiping away the stuff, which is what they do when it's a digging substrate. They have a very specific stance if it's to ingest something. So cool, we know that they won't eat it, but we know that they will bite through it. So even if statistically significant results aren't there, we still have these anecdotes that show us that, maybe we can do some more testing and better design things post-pandemic to be able to statistically show that these animals, that there is a difference or there is an improvement, a welfare improvement. But for right now, at least this research gives us hope. But there were some issues with it. Papercrete is great, but it's also so dusty. And we didn't know if, eating Portland cement or even just breathing it in because it is dusty is an issue. We don't know what it will do. We know that the newspaper will most likely be fine, but we don't know, will the Portland cement be fine? Because you're supposed to, when you cast Portland cement, you have to wear at least an N95. So if you have to cast it and wear an N95, what is the significance if, after it's cured, you're eating it? They're two very different compounds once they're cured, but there's still that concern. Also, it takes 28 days. So you have to plan 28 days in advance to be able to use the stuff. So we thought, was there something else that we can do? So what about just the paper? Because you take the paper and you squish it and it compresses and turns into a brick, as you can see here. So we took a chamber, a round chamber that we had, and I just cast the paper. I just squished it on in there, let it dry for maybe about a weekend, and it was pretty much fully dry. I tried to punch it through and break it, did nothing. So we thought, hey, let's give it a try. And Mikey liked it. So over here on this side is what we started with, and overnight they did this to it. And what was really interesting is they took the paper, and as you went chamber by chamber, you saw chamber was full of paper fluff, chamber was full of paper fluff, chamber was absolutely barren, no paper fluff, no paper fluff, no paper fluff, paper fluff, paper fluff, paper fluff. So they're using it differentially in their chambers throughout their colony, and in chambers that we thought had no purpose. Usually they designate one chamber for a toilet cage and one chamber for their nesting. But chambers that we hadn't seen them in, like in observation, they were using. Why? What does that lead to? What does that mean? What does it all mean? And so that's where we are right now, in that we kind of got the research back up in small parts and bits. Not a lot of researchers are allowed on campus right now. So it's really just Dan and me, which is a great start. But it's not the full swing and the full strength of the lab. So we're kind of still wondering how to do things and how to deal with logistics in a pandemic and how to adjust now to a more post-pandemic world. August 1st, we're supposed to get all of the campus back in full swing. So it'll be interesting to see what we can and cannot do. And we hope to continue doing research like this for these guys. And so that is my, and I will, leave this lovely picture for any questions. And I will also say that you're more than welcome to connect with me on any of these links, and I'll leave this up so that way you can find it, find me on social media. So yeah, thank you very much.
Thank you so much. That was absolutely fantastic. I've learned so much, and those Molerats are very cute. I need to meet them in person.
You absolutely do.
So if anybody has any questions, feel free to unmute yourself or just type it in the chat. And while we're waiting for that, I do have a few questions.
Sure. Oh my.
So first, I don't know if you mentioned this, but how did you decide to use Papercrete for the colonies? Was it just looking at the tiny home stuff that sparked the idea?
I mean, honestly, yes, a little bit of that. And, it was just kind of making lists of like, all right, what kind of material? So as a zookeeper, you kind of run wild with what you can do for your animals. In a laboratory facility, it's a little bit different because you can't just throw a tire in for your goats because it needs to be sterilized and it needs to be something that can be cleaned. So there's a couple more restraints. So I started with, the Zookeeper Pinterest board of what kind of materials would I love for them. And Dan is also this kind of researcher that he's like, I'd love to just fill a room with dirt and do it, just stick them in and see what happens. Which would be great in like a zoo setting, but not so great in a laboratory where we need to actually sterilize things. So it was kind of this process of, okay, what is the biggest dream? And then knock it down a little. What's more feasible? And then it became, okay, well, Anna also needs to be able to change it. People don't want to get dirty in lab. People want to be able to breathe and go out and do things after work and not be ostracized for smelling like a disgusting mess or something. So that's how it eventually came to be. It was just oh, well, this stuff is easy to make apparently, and it's there, and I love DIY, so let's do it.
Cool. And then we have a question in the chat. So it sounds like there are lots of really interesting fun facts about mole rats. What's your favorite fact about them and why?
Oh God, goodness. I think it's so cool that they have queens and that they have this social, so their social hierarchy. If they're one of the ways that we could track them is through RFID, which is I'm sure you guys all know this. I usually say it's like an easy pass. tag when I do this for like high school students. So they all have RFID chips in them, and we can track them with these rings around those PVC tunnels. And what they do is, depending on who's higher or lower in the social hierarchy, they will pass over or under each other. So the queen will always be on top. She always gets to pass over everyone else, and everyone else has to pass under her. And that kind of trickles down through the social hierarchy, that anyone who's higher up passes. And it happens like on cue all the time, no fail. So you can actually track these guys based on and figure out their whole social structure just based on who's walking on top of whom. And I think that's probably one of the coolest things. Also, the fact that their teeth come before their lips. It is something I had to get used to because it's a little disconcerting. But now that I'm like used to them, it's pretty awesome. I could go on about all of it is my favorite. How about that? All of it.
I mean, yeah, they're super awesome, especially that list of everything, the fact that they're cancer resistant. It's really cool. I like the saber-tooth sausages comment.
There's a more not safe for work in less formal literature that they call them. But that one is my favorite, especially because, you know, I'm sure everyone here knows about the charisma factor of animals and, you know, conservation, that the fluffy ones, the pretty ones are the ones that always get, you know, the most attention. The sea turtles, you know, the save the chimps, and that's really cool and great and all. But it's also these little guys that are doing things, you know, amazing for their environment, and are literally, you know, architects to their environment. And farming, they're farming things like they're sustainable, they're role models. And giving them a name like that, a fun name that they can attach to, is, you know, it takes the charisma factor up, and I really love that about them.
Great. So I was also going to ask, but then you showed that last slide. Have you tried filling an entire chamber with papercrete? And you did do that. So now my follow-up is, what is the idea? Like, what's the dream with this? Because it looks like you have to clean the chambers so thoroughly every couple of weeks. If a chamber is full of this papercrete, or you just give them a block to live in, how will that change the dynamic of cleaning? Is it safe for them to be living in?
Yeah, so that's a lot of questions that we, you know, we don't know yet. We want to be able to just have, you know, some giant block, because also what Dan wants to do with this is, in the same way that they will like put expanding foam or like melt silver into like ant colonies to see, you know, we're not going to be pouring silver on these guys, but we want to be able to look at what they choose to make, what, you know, what is the architecture, what is their architecture aesthetic, basically. And because that could give us more insight into what they want in their environment. And so we want to put them in this giant block, and then we would have to find a way to move them. So our USDA inspector was like, okay, how are you going to count the dead if there's any dead? How are you going to check up on them? How are you going to shift them out of this thing? And we were like, okay, that's point taken, touche. And, you know, we found, well, maybe they don't really like high oxygen. And when we open the chambers to take them out and to change over their enclosure, they immediately dart from the air. They like bad air, so they like high CO2. They'll occupy a chamber that has bad CO2. They'll run for oxygen. Okay, well, we can possibly just fill it with oxygen and they'll just push out to another side. Or what we can do is we can take an object that causes vibrations and stick it on one side of the chamber, and they'll feel that vibration and think it's possibly a, you know, elephant or something that is big that could destroy their chambers that they need to evacuate for. And that's how we could possibly get them out. So the dream is, give them a giant block of something to, you know, go through. So A, we can research them, and B, they could do whatever the heck they want in them. There's also ways that you can, you know, for birds, they have like the RFID like antennas around. So there's spikes that you could, you know, dig into things that we can be able to get more dimensions of information from them too. So that's what we're hoping to do. That's the dream.
And are you thinking, if that ends up being a reality, you would just need to use the RFID then to figure out what's going on, because you're not going to be able to see through.
Yeah.
Unless it's in an enclosed clear thing, but even then you'll only be able to see the tunnels that might be on the walls.
Yeah, so that's something that we've been thinking of. Right now, Dan mostly uses RFID to infer their behavior. And we have a giant supercomputer on campus that he uses to filter through that. I am not technologically literate in that way. So I did the more traditional, pen and paper route of just sitting there and observing them. So it leads to like, can we, infrared, like heat track them? What else can we do to see how they're moving and what they're doing? Because there is a lot of data and observations that you miss with just the RFID. Because sure, you can track them from where they're moving and they enter a chamber, but you don't know what they're doing in the chamber. They could just, if they're in the food chamber, are they eating? Most of the time they're not eating, they're just like laying around. So, how do we get that information? We don't know right now. Could it be just sticking like a camera in and seeing, what goes on, or embedding cameras in the material? But then how do you know, what chambers they're going to make? If they're going to run into a camera, will they eat through it because they have the capacity to do that? It's a lot that we just don't know.
Cool. Okay, so there's still a few minutes for questions. If anybody has any, put them in the chat for me. I'm trying to think if I have any more. I feel like I have so many. Let me see. So for now, I guess, are you still putting in the 2-inch blocks to see what they're doing with them? Or are you going to just use paper now for those blocks?
Yeah, so what we want to do is we've just used the 2-inch blocks, and the last picture was just paper. So what I want to do is I have all of those I just have giant blocks of Papercrete sitting around now for a year because I haven't been able to do any research with them. So the original thesis was going to be looking at like preference testing with them, of a concurrent presentation of all the different substrates that they have, and see, do they spend more time in one than the other and whatever. So we could still do stuff like that. It's kind of like Dan and I are both very, chill and like, what do we want to do? How do we want to do it? It's not really about publishing or anything. We're just trying to make sure that we do it right and do right by them. So we're kind of just like, what do we want to do? How do we want to do this? When do we want to do it? Who knows? Kind of thing. And it's also about like the logistics. Currently, as we speak, hopefully, Dan is getting in contact with a paper recycling company. And they compress their paper pulp into giant, like 500-pound bales. So we're going to see if they would donate 1 or 2 that we could test out. So that's what we're hoping for. We don't know if Christmas will happen early, but that's what we're on so far. I can tell you guys though about what happens if they escape, if you want to hear.
Oh yes, please talk about the escaped one.
So actually before I was working there, my then girlfriend was working at the same facility, and she took me on a tour to see the animals because I was going to be working there. And we opened the door and there's all of these Molerats that just managed to hop out onto the floor. And they can go forwards and reverse in the same speed and with the same dexterity and finesse. So it kind of just looked like a bunch of like K-turning cars just all over the place. And what happens is if they get out, the way that they recognize kin is through smell. So if they get out and they smell different, they will literally kill each other over it. So what you have to do when they get out, and this has only happened once or twice, and it's no big deal because it's only been a couple of them, and we just, plop them back in. You basically have to take these saber-toothed sausages and you have to find their toilet cage, because the way that they communicate who is kin and apply that scent is through rolling in their own feces. You have to take these sausages and you have to baste them in their poop and then put them in the colony and hope for the best with them, and obviously watch them and make sure. And that is what happens when they escape. That is our protocol, to basically baste them like a bunch of little chicken cutlets back into their colony.
Are they hard to pick up when they're running around?
They move very swiftly for these little rodents. And in other places, other facilities have said that they can't handle their Molerats the way that we do, because theirs are like super vicious. Ours, they're just like little like rice croquettes. You can just basically scoop them up, and they chirp. They make these like sort of guinea pig chirpy noises. But they don't really, unless they're already really stressed, they don't bite us. They just kind of okay, what are we doing now? I don't really like this, but here we are. And then you just kind of slather them and let them on their way, and they're chill.
How long have is it the same colonies that Dan has had for this long?
So we got our colonies from the University of Connecticut. I think in about 2010, Dan got a founding colony, and from that founding colony, we now have 7 different colonies. So what happens with these guys is what we think happens is that a princess kind of emerges. And so there's like factions that start to form, and the queen gets basically challenged, and there's like rivalry. So when we see that happening, usually we will separate the princess that we think is a princess, and take a male and put it with her, because what happens is they don't go through like a chronological pubescence or development. It's more of a socially like prompted development. So their puberty, especially for the queen, only once she's like coronated does she really start to go through those pubescent changes. A little before that, but mostly the changes happen afterwards. So if she's already like going through puberty, her chemical signals will prompt the male to also like kick into gear and go through puberty and go from being sterile, because all of them are basically sterile except for the breeders, to being a breeding male. And then we found a colony, and then we get to pick a scientist that we name it after. So I got to pick Linus, so we have Linus Pauling colony. So that's usually what happens. Now we have 7+ colonies.
Wow, that is fantastic. Okay, we're out of time now, but thank you so much, Jouvay, for joining us and for that really exciting talk. And for everyone who's here, if your friends missed it or if you missed part of it, this will be on the Zoology YouTube later. But thank you all for joining us today. And Jouvay, I hope you have a wonderful day.
I hope you all have a wonderful day. Thank you so much for having me.